Welcome to our deep dive into the fascinating world of proteins, and specifically how AI is changing the way we understand these essential molecules.
Yeah, it's a really exciting field, and this paper we're looking at today, multimodal pre-training for unsupervised protein representation learning, it really shows just how powerful AI can be in this area.
So the paper focuses on something called MPRL. That's multimodal pre-training for unsupervised protein representation learning. I know it's a bit of a mouthful, but it's actually a really clever approach
It
to get a
is.
much more complete understanding of proteins.
It really is. Think of it this way. Imagine you're trying to understand a complex machine, right? But you can only look at each part separately.
Yeah.
You might figure out what some of the parts do, but you wouldn't really get how they all work together. NPRL is kind of like seeing the whole machine in action,
Oh, okay.
seeing how the parts actually interact and work as one.
So to really grasp how revolutionary MPRL is, we should probably start with the basics of protein structure. I mean, I remember learning about this in biology class, but it's probably a good refresher for our listeners too.
Absolutely. Proteins are essentially chains of amino acids, which are like the building blocks.
Right. Yeah.
And these chains, they fold into specific 3D shapes.
Yeah.
And that shape determines the protein's function. It's kind of like how the shape of a key
Oh, I like that.
determines which lock it'll open. Yeah,
Yeah.
so the sequence of amino acids,
Living a blueprint.
it's like a blueprint, yeah. But the real magic happens when it folds into that 3D structure.
Exactly. Exactly. And that's where things get really complex, right?
They do. There are actually 4 levels of protein structure.
Four.
The primary structure is simply the linear sequence of amino acids.
Okay.
Then you've got secondary structures, which are local patterns like helixes and sheets.
Okay.
And these patterns combine to form the tertiary structure, which is the overall 3D shape of a single protein.
Okay. So that's three, four.
And finally, some proteins are made up of multiple chains that interact with each other, forming a quaternary structure.
Oh, wow. So it's like levels of complexity.
Exactly.
So going back to that machine analogy,
Yeah.
the primary structure is like the parts list.
Yeah.
The secondary structures are like, what, subassemblies?
Subassemblies, yeah.
The tertiary structure is the assembled machine,
Right.
and the quaternary structure is like multiple machines all working together.
That's a perfect way to put it.
I like that. I like that. Now, traditional methods for studying protein structure have always been really slow and expensive, haven't they?
They have. They often involve things like X-ray crystallography or cryo-electron microscopy.
Right. And those require a lot of specialized equipment and expertise.
Exactly.
But AI has the potential to really speed things up, right?
It does, yeah. And that's where NPRL comes in.
Okay, tell me more.
So instead of relying on just one type of data, NPRL combines 3 different representations of a protein. Its amino acid sequence, its residue level graph, and its 3D point cloud.
Hold on, let's unpack that a bit. What exactly are a residue level graph and a 3D point cloud?
Sure, a residue level graph basically maps out the connections between the amino acids in the protein.
So it's showing which ones are close together, even if they're far apart in the sequence.
Exactly. It's like a network diagram, kind of showing how the different parts of the machine are linked.
Okay, that makes sense. And what about the 3D point cloud?
Well, a 3D point cloud is basically a collection of coordinates
Mm-hmm.
that represent the positions of the atoms in the protein. It captures the overall shape, but it doesn't give you specific info about the amino acid sequence, like a 3D scan without any labels.
Gotcha. So MPRL is getting a much richer understanding of the protein by combining all three, right?
Precisely. It's like having 3 different perspectives on that same machine. You get a much more complete picture of how it works.
Now, how does it actually combine these different data types? Sounds pretty complex.
It is. It uses 3 different AI models, each one specializing in one data type.
Mm-hmm.
For the amino acid sequence, it uses a model called ESM2, which is really good at understanding the language of protein sequences.
Okay.
For the residue level graph, it uses VGAE, which is designed for analyzing network structures. And for the 3D point cloud, it uses PAE, which is good at processing spatial information.
So each model's like an expert in its own area.
Exactly.
But how do they all work together?
That's where autofusion comes in.
Okay.
It's a mechanism that merges the outputs of all 3 models into a single representation of the protein. It's like taking the insights from those experts and combining them into one comprehensive report.
So autofusion is the key to bringing all that information together.
Yeah.
Is there anything else special about the way MPRL works?
There is. One important aspect is that the models used in MPRL are symmetry preserving,
Meaning?
meaning they can recognize a protein regardless of its orientation. like being able to identify a machine, whether it's upright, upside down, or sideways.
That sounds crucial, especially with 3D structures.
It is, yeah. And the results are really impressive.
Yeah.
They tested MPRL on 4 different tasks that are commonly used to evaluate protein understanding, and it did remarkably well.
What kind of tasks are we talking about?
Well, one task was predicting protein ligand binding affinity, which essentially means predicting how strongly a drug will bind to a target protein. This is critical for drug discovery.
So MPRL could help us design better drugs.
Exactly. It can help us understand how changes to a protein structure might affect its ability to bind to a drug.
That's amazing. What were the other tasks?
Another task was protein fold classification, which involves grouping proteins based on their 3D shapes. Important because proteins with similar shapes often have similar functions.
So MPRL can help us understand the function of a protein based on its shape.
Yes, and it can help us identify new proteins with potentially useful functions.
That's incredible. What about the other 2 tasks?
So the third was enzyme identification.
Okay.
Enzymes are a special type of protein that speed up chemical reactions, and they're like essential for life. Identifying which proteins are enzymes is like crucial for understanding how cells function.
So MPRL can help us pinpoint these vital enzymes
Yeah.
out of all the proteins.
Exactly. It's like having a tool that can quickly identify the specific proteins that have these important roles.
That's pretty amazing.
Yeah.
Okay, what was the final task?
The final one was mutation stability prediction.
Okay.
And this involves predicting how changes to a protein's amino acid sequence will affect its stability.
So how mutations can lead to diseases.
Yeah, it's important for understanding that, but it's also really crucial for protein engineering.
Protein engineering, what is that?
So if we want to design new proteins with specific functions, we need to be able to understand how changes to their sequences will affect their stability. MPRL can help us make those predictions,
Oh, interesting.
which can guide our designs and lead to, the creation of new proteins with applications in medicine, agriculture, all sorts of things.
That's fascinating. So MPRL really excelled at all 4 tasks.
It did.
Showing it's got potential for a bunch of different applications.
Mm.
But let's go back to those 3 different AI models that MPRL uses.
Yeah.
In the first part, we talked about the residue level graph and the 3D point cloud. But what about that first model, ESM2? What's so special about that one?
ESM2 is actually pretty remarkable. It focuses only on the protein's amino acid sequence,
Okay.
which is kind of like, reading the protein's blueprint without actually seeing the finished product.
Right.
The surprising thing is that even without that 3D information, ESM2 still performed really well on tasks that you'd think would need, like, spatial understanding.
Well, how's that even possible? I would think you'd have to see the shape to understand how it works.
Yeah, that's what I thought too, but ESM2's power comes from the huge amount of data it was trained on. It's almost like a language model that has read every book ever written.
Wow.
By analyzing millions and millions of protein sequences, ESM2 has learned to pick out these subtle patterns and relationships within those sequences that give clues about the protein's structure and function.
So even though it's just looking at the sequence, it's somehow able to get information about the 3D structure.
Precisely. It's almost like it's starting to understand the hidden language of proteins.
Wow.
The evolutionary code that tells the amino acid sequence how to fold into that complex molecule.
That's incredible. So much information packed into such a simple sequence. Makes you wonder what else we can discover.
It really does. It speaks to the power of AI, not just to analyze data, but to actually reveal hidden connections and patterns.
Yeah, like AI is giving us a new way to see the world of proteins.
Exactly.
It's helping us understand these molecules at a much deeper level,
It is.
uncovering all these secrets of their structure and function and how they evolved.
And that understanding has the potential to totally change so many fields. Medicine, drug discovery, agriculture, you name it.
Okay, before we get too carried away with all the possibilities, let's take a step back and talk about some of the limitations. Are there any downsides to using AI like this?
Well, one of the biggest challenges in AI research, especially when dealing with these really complex models, is interpretability.
Interpretability, what does that mean?
It means being able to understand how the AI model is actually making its predictions.
Okay.
With some AI models, we can see the input and the output, but the process in between is like a black box.
Like a magic wand that works, but we don't know how the magic happens.
Exactly. And that lack of interpretability can be a concern, especially when we're talking about applying these models to real-world problems like drug discovery or disease diagnosis.
It's because if we don't understand how the AI is making its decisions, it's hard to really trust it, right?
It is, yeah. That's why a lot of research is now focused on developing models that are not only accurate, but also transparent,
Yeah.
so we can actually understand the AI's reasoning.
So we can be confident in its predictions.
Exactly.
That makes sense. So it's not just about creating powerful AI, but also understanding how it works and ensuring that we can use it ethically.
That's right. And that's where collaboration is really key, between AI researchers and domain experts and ethicists. We need to work together to make sure that these tools are used for the right reasons.
It's like we're explorers venturing into uncharted territory.
Yeah,
We're excited about the potential, but we need to be cautious and responsible.
we do. We're at the beginning of a new era in scientific discovery, and AI is playing a huge role.
Well, I'm really interested to see what the future holds.
Welcome back to our deep dive into the world of AI and proteins. It's been pretty amazing, hasn't it? Going from, the basics of protein structure all the way to MPRL and how AI is helping us discover so much.
It really has. And in this last part, we're going to look beyond this specific paper,
Yeah.
and explore some of the big trends and where this whole field is headed.
Okay, so where do we go from here? What are some of the next big things in protein representation learning?
Well, one area that's really exciting is incorporating even more types of data into our models.
We've seen how powerful it can be with MPRL, combining sequences and graphs and point clouds.
Yeah.
But what if we could add even more?
What kind of information are you thinking about?
Well, we could look at protein dynamics, for instance.
Protein dynamics.
Proteins, they're not static. They move and change shape over time. Capturing that dynamic behavior could give us like a whole new level of understanding.
So instead of just a snapshot, it's like a video.
Exactly.
Yeah.
Or we could incorporate information about how proteins interact with other molecules like drugs or other proteins.
Okay.
Or we could even look at their cellular context, how they behave within a living cell.
So it's like zooming out to get the bigger picture.
Exactly. And as we add all this data, we'll need even more sophisticated AI models to make sense of it all.
That makes sense.
Yeah.
Speaking of those sophisticated models, earlier we talked about that challenge of interpretability.
Yeah.
Are there any other challenges that are like, top of mind for researchers in this field?
One challenge is just the sheer volume of data. As sequencing technology keeps getting better, we're getting so much protein data faster than ever.
So it's a good problem to have, right?
It is, but we need AI that can handle it and actually extract meaningful insights from all that data.
It's not just about having the data, it's about understanding it.
Precisely. We need AI that can filter out the noise and find the real signal, the patterns that tell us something important about the protein.
It sounds like a lot of work to do, but with a lot of potential for some big discoveries too.
Oh, absolutely.
What are some of the applications that you're most excited about?
Well, drug discovery is one. It's already changing so much thanks to AI and protein research. By understanding how proteins and drugs interact, we can design therapies that are much more effective and targeted.
And we talked earlier about protein engineering, designing new proteins for specific purposes. Are there any applications there that you're particularly excited about?
One area that's getting a lot of attention is using protein engineering to create new biomaterials.
Oh.
Imagine being able to create proteins that could self-assemble into complex structures,
Wow.
like scaffolding for building tissues or even organs.
That sounds straight out of science fiction.
It does, doesn't it? But it's a real possibility. We could also engineer proteins to create new enzymes, ones that could break down pollutants or create biofuels.
So it's not just about understanding existing proteins, but about making entirely new ones.
That's right. And as we learn more about how proteins work, we'll get even better at designing and engineering them.
It's amazing to think how far we've come from those early days of, struggling to figure out protein structures to now using AI to not just understand them, but design new ones.
It really is. It shows how powerful human ingenuity is, our drive to keep learning and pushing the boundaries.
Yeah.
And who knows what incredible discoveries are waiting for us.
Well, this field is definitely full of potential.
It is.
And it's a really exciting time to be watching it all unfold.
It is. And to our listeners out there, stay curious, keep learning, and be a part of this amazing journey of discovery.
Because the future of AI and proteins is happening right now, and we can all be a part of it. Thanks for joining us on this deep dive.